Good afternoon. I'm Jen Koevary, the Chief Operating and Financial Officer for Avery Therapeutics. Thanks for your interest in Avery. I'm excited to tell you about some updates that we have. For those of you that don't know Avery, we're an advanced therapies company based in Tucson, Arizona, and we are leveraging induced pluripotent stem cells to generate functional bioengineered tissues and focusing on areas of large unmet medical needs. Specifically, we're a leader in the development of cardiovascular therapies. So our core technology is in the development of engineered tissues that we've specifically positioned for easy adoption. And a couple of attributes of these are the robustness of the tissues that allow us to deliver these using minimally invasive surgical techniques. So that could be robotic or VATS approach for reaching the surface of the heart, as in the indications that I'll be telling you about today. An advantage of this tissue engineering approach is that we can get broad coverage over the entire surface of the heart, and we can implant it using suturing techniques, which clinicians are very familiar with. It means that we don't have additional training that is required to implant this beyond what physicians are typically used to doing in the operating room. So that makes it very fast and simple for them to deploy and will be a technology that they want to use. Throughout the development process, we have paid close attention to the commercial feasibility of the end product. And a very important component of that is being able to have a product that is highly stable to allow storage and delivery. So with these tissues, we're able to cryopreserve them, and we can store them on vapor phase liquid nitrogen for months. We can also store them short-term for days or weeks at refrigeration temperature. So this makes it very easy to transport and to keep on hand as we're waiting for the procedure. There's no reformulation required, so it's very easy on the delivery system at the hospital. And we also have a very cost-effective and scalable approach, in that we're able to manufacture large volume of product in a small manufacturing footprint, so we can manufacture in the future hundreds or thousands of doses at a time. So today I'll be talking to you specifically about our engineered cardiac tissues, and here I'll give you a little bit more information on what these are. So we're using these iPS-derived cells. We have a partner iPiece that is generating GMP iPSCs for us. They're also presenting online, check them out. We're taking these iPSCs and generating specialized cells, in this case cardiomyocytes and fibroblasts. We seed these on a biomaterial and generate these functioning sheets of cardiac tissue. This is an image of one beating synchronously and spontaneously. This is the type of tissue that we would take and implant onto the surface of the heart. So the clinical opportunities that I'll be sharing with you today that we're currently pursuing are chronic heart failure. You're seeing a lot of that in cardiac cell therapy today. If you attended the panel that our CEO Jordan presented on today, a lot of people talking about heart failure. It's a big opportunity. A couple of others that we're pursuing that you don't see other people pursuing, at least as frequently, is ventricular tachycardia and atrial fibrillation. All three of these are really important needs where not all patients are being served by the existing therapies, and we believe we have a good solution that can reverse disease and improve quality of life. So I'll talk through each one of these in a little bit more detail of the specific populations that we're focused on and share some exciting data. So in heart failure, we're focused on patients that have ischemic etiology, so they'll be having a history of MI and seeing a cardiologist, getting put on optimized drug therapy. Even with the best drugs today, the disease over time does progress. Symptoms return. What we see is maladaptive remodeling in the heart, where the LV undergoes a remodeling process, increasing the diameter of the LV to try and bring on more blood, increasing myocardial mass to try and pump out more blood. Over time, the heart can't continue to compensate, in this way, and ultimately there's a 50% chance of mortality within 5 years of diagnosis, so a very deadly disease. At Avery, we want to interact with these patients in a broad spectrum of this disease and be able to send them on a new upward trajectory and improve quality of life. And we are working on doing that with our tissue-engineered product that we call MyoCardia. So we've tested MyoCardia in several gold standard models of chronic heart failure, and here are some data from a recently completed study we did in a large animal using Yucatan mini swine. So for this model, we use a 90-minute ischemia reperfusion, and following that infarct event, we allow the animals to recover for 4 weeks. During that 4-week period, the animals will undergo that maladaptive remodeling process that is typical of humans with heart failure. So we see establishment of scar, we see enlargement of the ventricle, we see increases in myocardial mass. We then treat these animals, and we've followed them for 6 months post-treatment using MRI to look at functional outcomes and using activity monitors on collars of all of the animals to look at quality of life measures. And what we see is that the untreated animals continue to worsen over time, so continual increases in the LV volumes in systole and diastole, continual increases in myocardial mass, whereas the MyoCardia-treated animals are moving in completely the opposite direction. We're actually seeing a reversal of the disease process, where we're getting a reduction in the LV systolic and diastolic volumes, a reduction in the myocardial mass back towards the normal value. And at the end of the study, the MyoCardia-treated animals have a lower total heart weight than the untreated animals. So these improvements in volume, improvements in mass, plus the activity monitoring we're doing, seeing improvements in overall activity after treatment, these are associated with improvements in survival and improvements in quality of life. So this could be a really exciting improvement in quality of life for patients and improvement in survival. We have done a lot of work in mechanism of action. This is becoming a more and more common question that we get asked, and we have studied it in vitro and in our in vivo models. And here are some pictures to help with the explanation of this. So in the disease state of humans and in these animal models with heart failure, in the infarcted tissue, there's this dormant epicardium and a scarred underlying myocardium. We're placing our MyoCardia tissue on the outside of that epicardium, and there's numerous factors that are secreted and interacting with that epicardium, activating that epicardium and rejuvenating the underlying myocardium. On the organ level, we're seeing that reversal in maladaptive remodeling and, importantly, an increase in myocardial blood flow. So this is something that we're still in the very early stages of studying. It is multimodal, and there are additional animal studies that we're doing and clinical trials, which will have the opportunity to study this in more detail. So the second indication that we're currently working on is ventricular tachycardia, specifically refractory VT, so patients that have been treated with drugs, ablation, and have a recurrence of VT. These patients have poor LV function and a very high chance of mortality. We use a model of chronic ischemic cardiomyopathy, and in a healthy animal, we see that there's a very good millivoltage amplitude and conduction velocity through that LV. In the animals that have cardiomyopathy, there is a worsening of that voltage amplitude and conduction velocity through the LV tissue. When we treat these animals and look at the electrical activity following 3 and 7 weeks post-treatment, we see that there is an improvement in the voltage amplitude and in the conduction velocity back toward the normal levels. And ultimately, we see a reduction in the incidence of VT. So this is really exciting. This could be a life-saving opportunity to treat patients with VT. And the third indication that I'll be sharing is atrial fibrillation. This is the most common arrhythmia in adults in the US, very difficult to treat, drugs and ablation. There is a high level of recurrence despite the options for existing treatment, and it poses a major risk of stroke. So with the model that we use, it's a large animal model in which we rapidly pace the right atrium, and it leads to persistent AFib, in which the animal is in AFib for several months. We see a dilation of the atrium that is typical of severe AFib, and we see a change in the RR interval, as you can see on this rhythm strip. So once the animals are in persistent AFib, we treat them with an engineered tissue on top of the atrium, and we get a restoration in sinus rhythm. And importantly, we've followed the animals for 6 months post-treatment and had no recurrence in AFib following treatment. So that's really exciting, especially in an animal with a dilated atrium. We also see a reduction in the size of that atrium, which is important. We follow these animals like our prior studies with activity collars and see a restoration in quality of life. So this is really exciting opportunity to treat patients that have AFib. Currently, we're in the process of planning clinical trials for each of these indications, CHF, VT, and AFib. We're working with 3 top-tier sites in the US and world-class PIs. We have a centralized imaging core, so all of the MRI and echo analysis can be done using standardized methods in one place. And we have been very attentive to the clinical feedback in designing these trials. So we're working with people that have a lot of experience in treating these patients and running clinical trials in these populations. All of these trials are going to be phase 1 trials, so our primary focus will be on safety. However, we have an opportunity to also collect information on function and quality of life. So we'll be using imaging and standard quality of life measures through surveys and things like 6-minute walk to be able to give us some information on efficacy that will also help us to design the phase 2 trials, which will look at efficacy. So that's what I have to share with you today. I will be around tomorrow, as well as our CEO, Jordan. We'll also be on the online virtual meeting next week, so please reach out to us if you'd like to learn more about Avery. We look forward to speaking with you. I guess I do have 2 minutes. Yeah. We are using an allogeneic tissue source, so we have a donor and we bank the iPSCs that we use to generate the cells.
And then I'm familiar with Terumo's heart sheet.
Yeah, so
what's your approach that's different?
Yeah, so the heart sheet uses a similar approach in that it is a sheet like ours. The major difference from what I know about Terumo's technology is the robustness of ours. So it can be picked up by hand, handled minimally invasively, so it can be deployed through working ports in robotic surgery. Another important aspect is that it can be cryopreserved, and so it's very stable to be shipped around the world. My understanding of the Terumo technology is that it is more fragile in nature and doesn't have the ability to cryopreserve. 1 more minute if anybody has another question. Okay, well, if you think of anything, I'll be around. Thanks for your attention.